So yes, thank you very much for that introduction. And hopefully and I'm sorry I can't be there. Of course, the magic of the internet means I can virtually travel much easier, but I'm sure I would have got a lot more by being physically with you. So I'm going to talk about sort of three technologies which are changing basic research and clinical practice, being genomics, imaging, and AI. Now, just to mention about the organization, which I'm actually second in command of, Deputy Director General, the European Molecular Biology Laboratory. We're Europe's only intergovernmental laboratory for life science research. We have these 5 missions to do excellent research, to perform scientific services or deliver scientific services, do advanced training for innovation and translation and integrating the life sciences. And in some ways, we're a sister organization to CERN that does high energy physics, or IISER that does rockets. We do molecular biology. We're headquartered in Heidelberg, Germany, and we have 6 sites across Europe. And the second biggest site is Amble EBI at Hingston, just south of Cambridge, which is dedicated to bioinformatics. And our new Director General, Edith Hurd, has kept us with one foot very firmly with the atoms and molecules and organelles and cells and aspects of living matter, but also stretched us to go out beyond the organism to how organisms work in populations and communities of organisms and entire ecosystems. And that's been a very exciting development. In some sense mirrors the way we reach out to human health, which I'll talk about more, and think about this as planetary health. But now I'm going to talk about these 3 different technologies, and I just would like to say that there's this wonderful review of Gregor Mendel, by Kim Naismith, and he made the argument that Gregor Mendel was the first biological data scientist. He went beyond observing nature to doing experiments and recording his data from which he derived principles. So, of course, his experiments were crossing peas with these different traits. What he recorded was the traits in the offspring, and he tabulated them to derive this kind of first law of genetics. And so in a similar way, I think we've been treading the same path as Mendel of rather than just one dimension of generating high dimensional, multivariate, multimodal data sets, and then applying not just tabulation, but all sorts of statistics to it, which come in different flavors, mainly in the time in which they were invented. You go from statistics to deep learning here. And that leads us to do 2 things. One is to generate models or understanding of the system. And the second is to make accurate predictions. So I'm just going to talk about the 2 major data gathering technologies we have. So the first one is DNA sequencing. And DNA sequencing has developed from the 1970s, where Fred Sanger created dideoxy sequencing through automations in the 1990s. And then the first kind of technology shift, of which the most impressive was Solexa, then brought up by Illumina, which changed the way you visualized or captured these DNA sequences from one-dimensional lanes on a gel to 2-dimensional places on a flat surface. And that allowed a huge scale up in the number of simultaneous molecules you could look at. Then on the right-hand side, I've got 2 different types of 3rd-generation sequencing. One are these very small pores, nanopores in a lipid bilayer, where DNA or RNA goes through that, and you can measure the deflection of current. And the second one, and that's from Oxford Nanopore, and the second one is from PacBio, which is about 0 wavelength polymerase sensing, where you only sense the incorporated nucleotides at the bottom of the nanowell using a quantum mechanics trick about photons and light. Just to say, I'm a shareholder and consultant to Oxford Nanopore, just as a conflict of interest. Now, over the last 3 decades, we've really improved many axes of the way we measure DNA. So one I've mentioned is the number of simultaneous molecules we consider. Fred Sanger started this with just 1 molecule. Some of the more recent machines will do 1 billion simultaneous molecules. We are going from the length of reads. Actually, the earliest Solexa reads were only 19 bases long. The longest Oxford Nanopore reads now are a megabase or longer than a megabase, so 1 × 10⁶ letters, bases. The cost has come down hugely by a massive factor of at least 1 million, and I suspect will continue to come down. The error rate has moved around. Some of Oxford Nanopore's early systems had a 1 in 10 error rate, which is tricky to deal with, but you can still deal with it. But Oxford Nanopore has improved a lot, and other technologies, as well as Oxford Nanopore, are closing in 1 in 100 to 1 in 1,000, and some technologies are in 10,000. 1 in 10,000 error rate. The time to result has moved from hours to months to hours, and the type of molecules we can sense is not just DNA, but we can sense both DNA and RNA, and we can sense modifications on DNA and RNA. And interestingly, there's no inherent biophysical trade-off in these axes. You could be on the outer edge of all of these axes. So there's nothing here that says that success in one axis means that you must give up something on another axis. And I think the future continues to be bright for DNA sequencing. Now, we in my own lab have done a bunch of different things, and my most recent work with colleagues such as Adrian Ledger has been focused on RNA modifications. And this is a recent paper which showed that we could robustly call RNA modifications. So these are changes in the RNA done inside of the cell, and often we don't quite know what they're for, but they're probably for different biological regulations, ways of the RNA being regulated. And what has this enabled? So one of them is the reference genome, and obviously the most iconic one is the human reference genome done in 2000. But these days there are ambitious plans to get the reference genome of, for example, all the eukaryotes on the British and Irish Isles. So there's a project well underway to get every single eukaryote on the British and Irish Isles. The other thing is that if you can move your molecular biology test, such as antibody pull down, so CHIP, or expression measurements, so RNA, and then these are called ChIP-seq or RNA-seq, then you have an incredible way to scale molecular biology, and that's been particularly powerful for single cell approaches. And finally, this is hitting the clinic and being used in clinical work. And I'll touch on this a little bit more at the end of this talk. Again, we have, in my own lab, provided some recent innovation to this, and this is my great colleague, Tom Fitzgerald, of looking at copy number variation. And if you're interested, I commend this paper. Tom has developed the most robust way to do association to copy number variation in the human genome, and that's given us new insights into genomics, genome-phenome relationships. So these are some examples. So the other methodology that I would like to stress is imaging. So imaging goes from the near atomic scale or the atomic scale with X-ray crystallography, which of course is a type of imaging, through to whole organisms with MRI scans and different types of optical techniques. And one of the really exciting things is really having a series of technologies that bridges this atomic to organismal view of life. And I'll come on to the incredible coupling of this with artificial intelligence, AI, to interpret these images. Now, we tend to call these things microscopes, but I want to stress that no longer is the human eye the data collection device. So all of these things have really quite sophisticated data collection devices, and a computer is needed to reconstruct this. This is at its most extreme, I think, in super resolution microscopy, where you only record flashes of light, little single photons that have flashed. And from the aggregation of flashes, you can reconstruct the position of molecules in space. And that position can be below the diffraction limit of light. In some sense, you're trading resolution in time to gain resolution in space. So I've got 3 examples here from my colleagues across EMBL. So the left-hand one here is from Julia Mohammed at EMBL Heidelberg, where she did an EM tomography, so electron microscopy, but in situ. So she flash froze cells, bacterial cells, some of which had an antibiotic and others which did not. And inside those sections, which she imaged with electron microscopy, she could reconstruct ribosomes. And she could therefore reconstruct ribosomes that had bound the antibiotic versus ribosomes that had not, and therefore know the in vivo configuration of the impact of this drug. It's really quite remarkable work. In the middle is work by my colleague Cornelius Gross and colleagues in Grenoble, EMBL Grenoble in France, of using X-ray imaging. This is a new technique of repositioning synchrotron radiation to get quite wide volumes of tissues, but at 100 nanometer or 50 nanometer scale. And what's exciting about that is you can really see subcellular or these complicated cellular structures, particularly valid for the brain, where connectomics is so important. And then on the right-hand side is an example of measuring using photons to measure a very different thing from the absorption of photons. And this is the interaction of sound waves, phonons, with photons in Boolean microscopy. And that gives you a measure of the stiffness of the material. And here my colleague, Robert Prevedel at EMBL Heidelberg was able to measure the stiffness of the growing zebrafish larvae. And you can see that the central notochord is different and stiffer than the surrounding cells, which are in orange. Now, both of these 2 technologies, genomics and imaging, I think are the sort of premier high-dimensional ways of measuring life. There are other ways, in particular mass spectroscopy, and of course there are some things which are literally measurements, you know, how fast did something run or something like that. But the dimensionality and the scaling of genomics and imaging means that you get so much more information from them. But we need a way of decoding that information. And a huge shift in biology has been the shift to AI or machine learning. So I think of this as really 2 pieces of technology coming together. So one is really good data engineering. Nothing in AI or machine learning can exist without good data sets to work from. And that's very much what EMBL EBI is about, and I'll touch on that more. But the second amazing thing has been the development of these kind of maths engines, such as JAX or PyTorch. And really what this allows us to do is write down sophisticated models, functions, which the engines will automatically differentiate. And so you can make the differential of this very, very complicated function trivially, and it will fit for data, do gradient descent very efficiently on GPUs. And that has allowed us to talk about models with millions, billions of parameters in the extreme, certainly hundreds of thousands of parameters, with us not having to really know every parameter and certainly not having to do the maths and coding for every parameter. So what can we do from this? And I classify what we do in three different ways: a kind of labeling-based deep learning, a modeling-based deep learning, and then alternative uses of this maths infrastructure. And I'm going to give you an example or examples of all three. And so just to mention this data engineering, this is very much what EMBL-EBI does. The European Bioinformatics Institute, we help bring data from around the world provided by scientists on publication, and we organize that data, and then we give it back out to the entire world. And we are the premier place in the world that does this. Very thankful for all the Indian scientists who do submit data to EBI or our sister institutes, NCBI in America or DDBJ in Japan, or PDBJ in Japan, the PDB US in America. And we aggregate that and give it out. And it's been a great feature of molecular biology that we do this. And then the right-hand side, this is not my field, but I'm an avid user of them, which has been the development of these incredibly sophisticated differential equations, where you can differentiate very, very big equations in these code frameworks such as JAX and PyTorch. So I'm going to give you an example of what I would call labeling-based deep learning, and that's where we take something and we label it with features that we know are biologically relevant. This is most obviously the case in images, and it's really transformed our ability to look at images. The left-hand case here is from colleagues in the Charing Cross Hospital, the Hammersmith campus in London, where they've been labeling the human heart, MRI scans of the human heart, and they can label the blood pool, which is in red here, versus the muscle, versus the left and right ventricle, and this can all happen automatically. In the middle is a growing Arabidopsis shoot, and this is work by my colleague Anna Krysiak in EMBL Heidelberg, where she can automatically label, segment, and cluster cells. And this has been extremely powerful in the way that we've interpreted cellular data. And what's interesting about her work is that there's less supervision is needed for this. And then the right-hand side is from colleagues at Oxford Nanopore, where the decoding of the signal, which is really a picoamp current going through the nanopore, is decoded by one of these transformer modules, kind of a transducer model in many ways. And that is the way you get base pairs being predicted. And that has been better than other modeling techniques. These transformer style models are very, very powerful. But we can go, I think, definitely AI has been more powerful than just this labeling problem. And we can actually come up with models which we really believe are meaningful for the biological problem. And a great exemplar is AlphaFold, the AI model developed by Google DeepMind, which predicts protein structures, and won the CASP competition 2 years ago and started a complete revolution in protein structure prediction. Similar to this is the ability to predict tracks from DNA sequence, so to interpret the genome, and a number of people, including people at DeepMind and in Stanford, are very successful at this. And then the right-hand side is the ability of fusing multimodal data, in this case genomics and in situs of cancers, to derive features of the cancer, and really to think about these spaces independently of, jointly between genomics and histopathology. Just want to focus on AlphaFold as an example. So AlphaFold would not have been possible without the 40 years worth of patient aggregation of experimental data from around the world via the worldwide PDB. And in Europe, that is run by PDB-E, which is in EBI. Really impressive data engineering by my colleague, Samir Valanka and others. Of course, much of the smarts of this was the impressive end-to-end scheme of AI, of AlphaFold. John Jumper in DeepMind deserves huge credit, along with Demis Hassabis, of course, for doing this. And it's interesting hearing them speak about this. And one of them, of course, is the ability to write these models down as an end-to-end process. And then they train the entire system as one very, very complicated gradient descent. Now, when they did this, they came actually back to us, myself and Samir, and said, we want to make AlphaFold open. We want to make the code open, we want to make the method open, and we want to provide the results of running this AI. And they partnered with us at EBI to make the AlphaFold protein structure database, where we have stored the results of over 200 million protein structures. This emphasizes that when we can solve a problem using computational methods, we can scale that in a remarkable way, in a way which is really hard to understand for other techniques. Then my third example is using the same maths infrastructure, but for a different task. And just to say, the maths does not have to be neural networks. So this is an example of my colleagues, Maritz Gersten and Alex Jung, along with Kumar Gururav in my own group, doing machine learning using, a classic 1970s model of epidemiology. And we have modeled, or Alex and Kumar have modeled rather, the entire Danish National Patient Registry for cancer and for other non-cancer diagnoses. And that has allowed us to make the most accurate prediction of cancer risk using a total healthcare viewpoint of this. This is some of the results of that, and they ran this, or Alex ran this here in both Denmark and in the UK. What is interesting for people who do epidemiology here is actually the models we created were very transferable from Denmark to UK, far more than we were suspecting. Not every cancer was that. For example, the liver cancers, where it's a lot about alcohol abuse, that did not transfer well, and we kind of understand why. And Denmark is the entire population, whereas UK Biobank are volunteers and people who are susceptible or drinking too much basically tend not to volunteer for these things. And this means that we can do risk prediction significantly better than just age and sex for all different types of cancer. And that means that conceptually, for the same conceptual screening cost, we could increase the number of cases identified, and on average, they would be 1.5 years earlier, and 26% of cases would have been found before the screening would have started if one developed, applied this method as a screening method. Now, this is, of course, a proof of principle. I'm not suggesting that this can be rolled out as part of a screening process now. There are many, many other considerations for rolling things out into screening. It's not an easy topic. But it shows you that we can make real benefits by bringing big data and AI or large-scale calculus engineering to these problems. So I hope you agree that we would like this future to continue and to grow. And so what do we need to do? So the first thing we need to do is we need to continue to store and organize fundamental biomolecular data in an open way. Things such as the PDB and the DNA database, INSDC, the proteomics database. There's a modern buzzword to that. So this has an old history. It starts in the 1970s. There's a kind of 2000s buzzword called FAIR, findable, accessible, interoperable, and reusable, which is also totally applicable even to this data from the 1970s. And one way of thinking about this is we not only need data scientists, but we also need data engineers. So these are people who have an engineering mindset, but it's about flowing, moving, curating, providing metadata, and organizing data for people to use downstream. Now at EMBL EBI, this is what we do. Scientists generate data and make discoveries. They deposit it with EMBL EBI in publication. And again, many thanks for all the people who have done that. We archive it and share it with global collaborators and all scientists. Everyone can download everything that has been shared with us. We classify, enrich, and combine and analyze it to make easier to use data sets, such as the Ensemble browser or the Uniprot data set. And then we distribute both the raw data and the value-added data resources. I should add there's one exception to you're just allowed to download everything. For data which has been shared from human subjects with a specific consent, you must be authorized by the original data access committee to get access to that data. But that is a minority of the data, though, of course, it's incredibly important. And then we hope that that gives scientists new ways to design experiments on the basis of shared global knowledge. This is a picture of an average day at EMBL EBI. You can see it's very busy, and you can see there's a lot going on in India, as well as everywhere else in the world. This is being coordinated at a high level globally, and if there's a funding agency in the room, I really encourage you to join the Global Biodata Coalition, which is really a way of funders worldwide coordinating this. Sadly, there is not the UN of Science or something like that that coordinates it. So this is the next best thing, or perhaps even better, in the case here of biodata. So please participate in this. But there's another really exciting thing, and that is that our science, molecular biology and imaging, has become really useful for healthcare. And I'm now going to give you some examples where we can use healthcare, enable healthcare to happen using genomics, but also benefit from healthcare using genomics. And that in healthcare parlance, this means secondary use of data. So this is secondary use of genomics and imaging data from healthcare. And my best example here is Genomics England. This is the Department of Health in the UK's dedicated company for delivering this. And I have the privilege of being a non-executive director. So I'm part of the governance of this organization. So this is an example of where we're doing something. EMBL EBI is an example of a data commons where we can share data very openly. But Genomics England is on the right-hand side of this, where you cannot share data because it's really about too many people and too much detail. But what is acceptable is people coming and virtually visiting the UK, just as I am virtually visiting here India, but in this case using virtual machines and cloud resources based in the UK, which you could get access to, for example, from India. To make that future real, we have instituted another global standards organization to make the standards as you move between the UK and Japan and France and India and America as aligned as possible. And this is the Global Alliance for Genomics and Health. I'm the chair of this organization, and we're having a plenary in San Francisco this September. So please do come along, and it's about standards in this secondary use of healthcare data. This is a list of all of those standards and how we go about it. So does this actually work for healthcare? Oh, for healthcare and for science. So I'm not going to spend a lot of time on how AI works for basic research. A really great example is the nuclear pore from Jan Kaczynski in EMBL Hamburg of integrated structural biology. But I wanted to go to these clinical operations where we're having a real impact in the clinic. So this is data, the most recent data from Genomics England. There are about 50,000 live births a month in the UK. And about 2% of those have a possible genetic disease, often recognized within the first 24, 48 hours of birth, often, and then in the next tranche within the first 6 months to 1 year. So very, very early on in many people's lives is a clinician concerned about a child, and it might be a genetic cause. So that's about 1,000 probands, so people with concern. And these days in the UK, those individuals, often children, can have their genome sequenced by Genomics England. And wherever possible, we also sequence their parents, obviously with their consent. And it usually means there's about 2.5 genomes per proband, which is at the very least child and mother. So this ends up being about 25,000 genomes per year. And we get a diagnosis rate of about 25 to 30% of those children can be accurately diagnosed using genomics, from their genome. And this has a big impact. So on average, overall, diagnosis leads to 50% less hospital visits from the child, which is a big saving to the child, less painful, and it's also less costly to the healthcare system. Around 5% of patients have a large transformative impact on their future care, and I'll come on to some of the best cases of that. And finally, the family is able to make better informed choices for potential future children. And then, as I mentioned, this is now closer to 90%. Around 90% of families consent to ongoing research. And so that's the secondary use of research for the benefit of secondary use of healthcare data for the benefit of research. So I just want to give you some examples of profound changes in outcome. Probably the best is the RPE65 gene. This is a gene which, when knocked out, will nearly always lead to the loss of vision. It's a gene in cells in the retinal pigment epithelia at the back of the eye. And there is a gene therapy, Lux-Turner, which will cure that. And the cure looks very long-lasting, at least 10 years. And so these AAV vectors with the transgene are injected, they integrate into the retinal pigment epithelia, and these children don't go blind. It's about 30 children a year in the UK who now get this treatment. And just to say the obvious thing here, it's completely dependent on accurate diagnosis. If you are not able to diagnose the effect or misdiagnose it, then this method won't work. This little boy in the middle here is an example of CTPS1 deficiency. This is an immune system deficiency. Now, very severe immune deficiencies are usually recognized within 6 months, but there's sort of sliding scale of slightly milder immune deficiencies. And this child came in for its 3rd round of chickenpox when he was about 5 or 6. And that really concerned the doctors, and that's not normal. And so they sequenced the genome and discovered that he did have a deficiency in this particular gene. And so the doctors scheduled him for a dangerous well, not certainly non-trivial treatment of a full bone marrow transplant. But that was successful, and the child now is expected to live a healthy life. A final example is maturity onset diabetes of the young. These are a type of diabetes which you can diagnose very early in life. And if you treat them with the right drugs, then it has very little impact on the future of those children. But yet again, you have to match the right drug to the right diagnosis for it to work. So I hope that's been interesting. I've used work from my own research group, and there on the right there in lovely sunny Northumberland on a retreat. And I've also talked about work from the Danish Healthcare Registry, and that is my set of colleagues across Denmark and my own research group who've worked together on that. So I'm very, very happy to take questions.
Thank you very much. Questions? There probably are. So I have a question about, you mentioned that the one data type that's not freely available is human data, but that's more and more relevant, and it's been a topic of discussion at this meeting also on how to de-identify human data, how to anonymize it, how to use it via mechanisms like federated learning and so on. So can you expand a little about?
I mean, just to say, I think a lot of human data can be accessed, but you have to go through more steps. And so that, we call that controlled access in our terminology. But the trend here is towards sort of walled gardens, so private cloud installations, where that data is accessible, but for the people who are allowed. So a great example is Genomics England. I have access to the Genomics England data set on their controlled private cloud, but I cannot download it. And if I want results, I have to go through a process to show that the results I've got are not identifiable. So that's a kind of step before federated learning. You could call this remote access or cloud or other things, but it's very practical and very usable. And I think that's a very important step. I do think federated learning techniques are useful, and I also think that secure computing is a useful concept, and we should do the research to allow us to do federated learning. But I want to stress that you can do an awful lot with just cloud-based access. And then, I mean, there's this kind of question about whether you call meta-analysis federated learning. In some sense, it is. Meta-analysis where you combine the results from different, say, from France and Denmark and England, where you don't combine the raw data, but you combine the results in a meta-analysis. And there's, of course, a long statistical history for that, so of doing meta-analysis between studies.
Any other questions? Okay, so I had one more question. This was about the, you said you want to sequence possibly every eukaryote in the British Isles.
Yeah.
And that includes insects and, invertebrates and so on, because there are literally millions of those. Is there a targeted,
Yeah, so actually, for all the multicellular eukaryotes, which I know is a bit of a weird thing, so all the multicellular animals, plants, and fungi, there's about 70,000 different species. So obviously quite a lot of insects, an awful lot of flies and butterflies and stuff like that. But it's not crazy. And they are making good progress. I think they're doing 2 a day at the moment. So they will make it, I think, by the end. Protists, so single cell eukaryotes, we don't really know how many species there are, and some people are going after that. And obviously, across the entire planet, maybe if you think about those multicellular organisms, I think people think there's about 100 million of those, which again is quite a large number, but it's a countable number. So obviously doing it in the tropics, life is that much richer in the tropics. But I think it would be fascinating to do it in more and more places.
All right, so let's thank Professor Birney again for bird's eye overview of what API is doing and what are the future possibilities. So thank you very much and see you again on Friday.